Let's get started. Good afternoon, good morning, good evening, everybody. Thank you very much for joining us. My name is Kerry Langley, and I look after the commercial side of VSN International. Presenting for us today will be Dr. Salvador Gazan. Apologies on my pronunciation. Salvador is our resident statistical geneticist, mountain biker extraordinaire. Just a quick bit of housekeeping, I'll keep it brief. We've got quite a large audience today. I'm hoping we might have a little bit of time at the end for some questions. Do feel free to post in the chat. We'll pick up where we can. Always nice to see who's here, where you guys are from, and what you're hoping to get out of today. We will be sharing the recording after the presentation to everybody that's registered, and also do please feel free to share that with your teams and your colleagues. More than welcome to do that. Yeah, we'll also send an email following the presentation, which will give you contact details should you have any specific questions on our products and services, and also some exclusive free trial offers, so you can have a chance to demo our products and services. Yeah, as I say, that's it. But do feel free to post in the chat, Amy. I think that capability, that feature is turned on. And for now, I will hand you over to Salvador. Please excuse us if we turn our video off, because we're having some minor connection problems which seem to improve when the video is off. So please bear with us. Thank you. Over to you, Salvador. You're on silent.
Thank you. Hello, good morning, good afternoon. Nice to have you around. I know some of the participants, and it's nice to have you in this webinar. So again, as Carrie says, any questions, things, you can use the chat there. You can also contact me to that email. And if you have anything more about licensing and other products, also me or directly to the support. So I'll just go directly to some elements of this seminar. I'll try to keep it within the hour, but again, please feel free to kind of like questions and doubts and things like that that you might have, because this is obviously a relatively recent topic with some excitement. So the title is a bit about combining pedigree and genomic data, and it's focused on my experience in plant breeding. Again, this not only applies to plant breeding, but applies also to other fields. But it's specifically relevant for plant breeding, and I worked in several tree breeding programs where some of the use of the pedigree was more relevant. And we will be using GBLUP analysis, which is where we focus, that provides us flexibility to do more complicated and flexible analysis. So the first thing I wanted to start is what I'd like to call this like triad, the phenotypic data, genotypic data, and pedigree data. But let me start with the pedigree data. As we know, and I'll go quickly with this, the pedigree it's been one of the most important elements in animal breeding, and it's good to keep track of the pedigree because obviously keep tracks of this relationship between individuals. And in some breeding programs from animal breeding, you have thousands or maybe even millions of individuals with their pedigree, and most of the improvements we see come from keeping track and using this pedigree to track down relationships. Now, I noticed that several breeding programs, particularly in some of the agricultural crops, they do have pedigree, but the pedigree often is ignored, and this is fine. But obviously, because the focus has been on clones and total genetic values and other elements. But again, that's an important source, having the pedigree and using it. So that's what I wanted to talk in this webinar and bring that on. Now, there is one of the things that tends to happen, and this you will see when you use genomic data. In the genomic data or genomic analysis we do with GBLUP or another models-based B or whatever you're using, we obviously focus on the individuals that are being genotyped. And this is a bit of a problem, but the problem comes because obviously we currently have for many breeding programs lots of information on pedigree data for several trials, several individuals, but the only thing we have from those individuals is the pedigree information. So that has been a big investment, and we cannot just say, oh, I'm going to toss it away because we don't have those individuals' genotype. So for that reason, we need to kind of have a different alternative to use. And I'm not going to say anything new because everything's in the literature, but I want to illustrate and present a case where we'll bring this specific pedigree information, combine it with what we have currently of genotypic information, and then get a more robust analysis that uses everything that we have. Because my philosophy is that we shouldn't be dropping all data that we have, and this is going to be even more relevant as we get a better hold of genomic analysis. So you're probably aware where I'm going to. I'm going to the GBLUP analysis, and particularly the single-step-GBLUP, or some people are calling it HBLUP for hybrid BLUP, and I'll walk you through it a little bit. So again, just a kind of just a very brief overview of the pedigree relationship matrix. Again, we use often a pedigree file, and we record the individual and the parents. When we don't have information, we put a 0, and we know the parents, we indicate which ones are the parents. And then we go from the kids to the parents, the grandparents, great-grandparents. So it's not uncommon to have 8, 10, or 15 generations of data in some breeding programs. Now, when we use that pedigree file, depending on the software we use, we construct an A matrix, which is the numerator relationship matrix using Henderson equations that we are all kind of familiar. And it calculates some identity by descent or some relationship between individuals. And you have on the right here an example where you can see values bigger than 1 in the diagonal, what represent inbreeding. Hopefully all of them are 1. And depends, the maximum value is 2. And then in the off diagonal, you have the relationship between individuals. Like, for example, 3 is a kid of 1, and some individuals are related through parents that are related. So they have a relationship that is bigger than 0.5, and so on. So it's very good because this correlates everything with everything and allows to make this analysis and obviously predict breeding values. And we use our famous, and I say famous because the animal model has been around a while, but now with the emergence of all these genomic tools, and particularly with the GBLUP, we have like a resuscitating of the animal model where we obviously estimate a breeding value for each of the individuals that we have in the pedigree if we are using this pedigree-based relationship matrix, or alternatively, we replace that matrix by a genomic matrix, which I'm calling GA, which is based on the genotypic data we have, and then we call that GBLUP or genomic BLUP. So it is a very simple model in the sense that uses that relationship matrix to estimate the effect of each individual. As you can see here, we can have several fixed effects, several random effects depending on the origin of our data. We have an experimental, or even more complicated models if we have some heterogeneous structures and so on. So that is really our key model. Again, remember, it is to estimate breeding values that we can call GEBBs for genomic breeding values, or just EEBBs when we use the pedigree matrix. Now it comes the genomics data, and this is where we calculate our genomic matrix, that's one of the names, but we know also has another name, observed relationship matrix, realized relationship matrix, and some people just call it G matrix instead of A matrix or GA. So you can see it in different names. Now, one of the things that it is relevant on this matrix, obviously, it is going to be an approximation of our A matrix, where we have relationships that we know with expected values. For example, in this case, this might be a kid. This will be half-sieves. So when we genotype the individuals and calculate this realized relationship matrix, we are actually using an estimation of that relationship based on some Mendelian sampling that happens. So that's not going to be identical, and some other issues that are associated with this molecular matrix. But this is a very good approximation to A, and it has very good information. And both of them actually can feed into each other to improve our analysis. And again, that's a little bit of what I want to present here. when we use this genomic matrix, it's not free of issues. So one of the first decisions we need to do is what calculation of this genomic matrix we are going to do. And we have the most typical one that you probably have heard is Van Rayden. It is very common in plant breeding and in animal breeding. And there is another one, Yang, that is common in human genetics. And there's other definitions too with the slightly different variants and so on. So it's an ongoing topic of how to calculate this. One of the good things to remember is that even with different ways to calculate it and slightly different parameterizations, you will still end up with a very similar result analysis. That means the breeding values or the GBBs are going to be very similar regardless of this matrix. However, when you calculate this matrix, it's unstable and it needs to be inverted to be used on the mixed models equations, and often has some issues. Some individuals are highly correlated, more than what we'll expect. We have sampling errors to make the matrix unstable, and then therefore we cannot invert. And we have to do some manipulations and so on in order to make it stable. Again, those manipulations often change very minimum our GEBBs that we are going to calculate at the end. But it's important to focus on what happens on this matrix to assess if there is some issues. And I'll come back to this topic about getting the inverse of that matrix because, as I mentioned, the mixed model equations require the inverse of this GA matrix or the A matrix. The A matrix is easy to invert, but the GA is not so easy to invert. And it was also full of information and relationships. And again, sometimes has a determinant close to zero, which creates some issues to put everything into the mixed model equations for solving for the breeding values. So we will, I'll show you a few little tricks and things to manipulate this matrix and so on, alignment, bending and blending. And there's many other ones. If you look at some of the literature, you'll find slightly different approaches, but they all focus on the same or how to tweak this matrix so it is invertible. And I'll be using the library that we built at VSN. This is the library ASRgenomics, and you can download it from here. It is not the only library you can use to manipulate, but I'll be using it to kind of illustrate what the philosophy is. Where do you go when you have some problems? Some of the reports help you to come back and fix issues. So it just gives a little bit of a workflow that it's kind of nice. I use it a lot for consulting, for teaching too, and it is a really, it just puts together all what we thought about how to manipulate these things. So I'm going to go back to, I'm going to go to my code in R, which if you will have access to this code too if you want to. So I'm going to just get started. Hopefully you can see my code fine, no problem. So I'm just going to walk through it and show you a few things and how things are working, okay? So the two libraries we need is ASRgenomics and ASRemel. ASRgenomics, it's a free library that we have, so you can download directly from our webpage. We have free software, and you have ASRgenomics, and we have other ones and other more to come. And you can do everything you need, but the new beauty of it is it prepares some of the elements directly to be used in ASRemel, which again is our commercial software that you can also find and download and request license and so on if you want to use our product. But again, you don't necessarily, with this webinar, you can use the same tools for whatever libraries you normally use, but I'll use those two to illustrate. So the first thing I want to show you is, after this kind of like a review, this is a typical data that we have for pine. And actually, I'm going to put question mark phenopine. This is data is directly in the library that are ASRgenomics. So this corresponds to individual deregressed breeding values, and you can think about adjusted phenotypic responses for diameter at breast heights from trees from a specific site, in these cases in Florida, and we have 861 genotypes that have been phenotyped, and we are ready to do fitting a genomic selection model with this data. Now remember, it's 861 individuals, so I'm going to put that one here, 861. So I'm going to just load that data and show you a few things. And what we have is we have the third generation of individuals, and I know that because I work on the similar portion of this data. And I have some information about the mother and the father, and I have this adjusted or deregress breeding value. Think just like a phenotypic response that is being corrected by everything is the one I want to use to fit my model. Because it's centered on 0, you have some negative values and positive values. So when I go to the tail, you see I have some positive and some negative values. So don't worry too much about it. It's being just centered data. And as I mentioned, the dimension is 861 individuals that have been phenotyped. Okay. On top of that, I have my pedigree data. Now the pedigree data, and this is the third generation. And again, this is what I consider very important for many breeding programs. Sometimes you're in the 6th generation, but you're only being genotyping the last one or the last 2 generations. But you have all this historical information that you're using on a pedigree analysis or on a clonal analysis, if you only do clonal, but you have that valuable information. And suddenly we have this genomics data and genomic push, and we say, I'm not going to use it. I don't think it makes any much sense, because when we have the parents or the grandparents' phenotype, they do contribute to the breeding value of the performance of the offspring from which one we are doing selections. And it also might be that some of the parents, now that we have more data on their offspring, it's outstanding and we want to go back and select it, and it's not being genotyped because maybe we have it in a bank or something. So this is the pedigree, and the pedigree, this is just a few lines of the first generation, and on the tail, I'm just showing you a few lines of the last generation. So this is the third generation. These individuals from the third generation are the ones that are being genotyped, not all of them. Now, the dimension of the pedigree is 2,000 records, and this is relevant because it's 2,000 records that I have, but remember, I only have phenotypic data for a total of 861. So I'm going to put down here 2034. And that means I have a lot of more information on the pedigree for parents and grandparents that I want to use. And finally, I'm going to go to the molecular data. The molecular data, it has some description here, is only 655 genotypes. And this means out of the 861 individuals from the last generation, not everybody was genotyped. This could happen for many reasons. You just subsample, or something went wrong, or the individuals, something went wrong with the genotyping, or you just didn't have the resources more than just to do a subset of them, or when you do the quality control, many of them drop down and then you can't kind of eliminate them. But don't forget that even if it's 655, we have phenotypic information for 861. But we could even have more phenotypic information if we added the parents and the grandparents. So we could eventually have 2,000 individuals where they have phenotypic data that can contribute to the 655. And this is the main thing I want to point out, that there is more data, and when we just say, oh, I'm sorry, we only have 655, and we drop everything, it's inefficient. It's a waste of money. It's a waste of good data. So the data is here. I just show you a few lines. So we have this coded as 0, 1, 2, and actually we have a total of 4,853 markers. It's not too many markers. It's good enough to do what I'm going to illustrate. Again, depends on where you are. You might be talking about 20,000 markers, 50,000 markers, etc. But this is a reasonable number for what I'm going to illustrate. It's a reasonable number to get a genomic prediction model for this species. So I have 655 records, so you see here coded 0, 1, 2, and the missing values in this particular case are not coded as NA, are coded as -9. That's how it comes from the original publication. So I have an idea of the data, the dimension, and I'm going to use ASRgenomics directly now to facilitate my work. And again, this is one of the things I wanted to show you. This QC filtering function does a lot of little filters, facilitates that you can filter by individuals that have more than 20% missing, like in this line here. markers that have more than 20% missing, any markers that have less than 5% minor allele frequency, some level of heterozygosity that you want to, yes, too high heterozygosity, you can drop them, etc., etc. And so it facilitates some of the work. So just kind of automatically. There's other things that we have here. You can do a simple imputation. This is not a fancy imputation, just as the mean average imputation. You can do this filtering and then impute with your favorite software, Beagle or FImpute to do your imputation. So I'm just going to run this. And the nice thing is we thought about different, let me make this a bit bigger, different reports. So there's this number of markers. Some values were identified as missing values. 156 markers were dropped because they had too many missing. 11 individuals were dropped because they have too much missing, and so on. A lot of markers were dropped because of the MAF, which is not unusual, markers with other problems. You can play with this. At the end, we have 237% of missing cells, in this case, markers that have missing information on their SNPs. And we have our final matrix of 644 and 3,000 markers. So already I lost 11 individuals because their quality is not very good. These objects within this object is the clean matrix and imputed, if we ask for imputed, otherwise it still is a -9 or NA. This is the dimension, and we have some plots here to help us assess if we want to change. So this is individuals with high levels of missing, so we can change that to 30% if we want it. This is the missing on the SNPs, some SNPs. So again, if you have very high levels of missing, you will say, okay, there's something going on my data. Something similar with the minor allele frequency. We have lots of them that are being fixed on this population. That's why we drop so many. So little things like that can help you to kind of assess heterozygosity, and finally the inbreeding also within marker, just to see if you have something that it might be a bit weird or markers with some issues. I do recommend you filter this adequately because obviously bad data will eventually lead you to problems downstream, to get problems on the genomic matrix and so on. So I'm going to move to get my pedigree matrix, which is the A matrix, or sometimes they call it kinship matrix. I'm going to use this library AGH matrix, which is in CRAN, and we use a specific function, which is the A matrix, and reads the pedigree. Again, I'm assuming the pedigree is okay. It might need some checking, and I'm showing you here just a few lines from this pedigree. You have values of 1 in the diagonal and 0.5 for some of the offsprings that happen to be full SIPs. There's some 0.25s and so on. So this is good. We can assess this a little bit more if we have a complicated pedigree, check for mistakes, etc. I'm not going to talk too much about it, but it's important to make sure you can trust your pedigree, and sometimes you cannot. So in ASRgenomic, we have the function G matrix, which is actually going to calculate the genomic relationship based on the markers. It has options for additives and dominance, and again, it has some little reports that help us to see how good this matrix is, and you can also generate it in a sparse form for ASRemo. So this one, depending on the size of the matrix, might take a bit longer. Again, I have a 644 by 644. Remember, we're losing 11 observations. And then now we have this matrix here of relationships, 0.94, 0.93. Again, there's many ways to calculate this. Values that are close to 1 in the diagonal, many values of 0 on the off diagonal, and there seems to be some mild relationships, 0.07 and so on, 0.16 in this particular case. Now, if I compare this one with the A matrix, which we'll do a little bit later, we can see where some inconsistencies occur, and that's important. But before I move farther with this matrix, I want to assess the quality of this matrix. And this is done with this function kinship diagnostics, where I have several options, but the main thing is that it's going to report elements on the diagonal that are too high or too low, and also potential duplicates, and in this case is with a correlation of 0.95 from that genomic matrix. It's very important to check this because Inconsistencies will happen later. Sometimes you have individuals that are duplicates, and then they will be reported here. Levels of inbreeding that you didn't suspect, which again might denote issues on your genotyping, and so on. So there is some reports on the ranges. So for example, the mean of the diagonal values is close to 1, which is okay for this population. We have the whole range, 0.84 to 1.10, so that's not bad. And then values on the off-diagonal go all the way to minus 0.16, which is a bit wide range, and to 0.67, which is also possible for this population, and it doesn't go very high. And the mean is something. No reported individuals that are outside of the diagonal of our range, reported individuals that are potential duplicates, but if you find duplicates, you should go back and try to assess why they are duplicates and eliminate one of them. Again, just a graphical output of the values on the diagonal, or we can request a graphical output for the values on the off diagonal where there is something interesting that I wanted to show here. So I'm going to make this one a bit bigger. So I want to use this one again to combine with a pedigree matrix. And notice that you have individuals on the 0, which are unrelated on the off diagonal, but this is the half sieves, and this little lump here is the full sieves, and you can see there is a bias. This lump should be around 0.5, and this lump should be around 0.25 for this population, which is only 3 generations. So obviously we have some bias produced by the calculation of this matrix, and we can do different things. And that's where I was talking about the alignment. So when I go back here, and tell me if you can see my slides, we can see that we have an alignment. What we'll do is we'll match the G to the A. So we'll move the G, so some of the values of the G match the values of the A, for example, equating the diagonals and be a similar scale on the off diagonals, okay? And there is some references and more information. So that's what I'm going to do at the beginning. So the function gTuneUp will do that. And there's many other things, because in the function gTuneUp, we also added bending and blending. So what I'm going to do is, look, I have the A matrix with 2,000 records. The G matrix, after my filtering, is 644 records. And with this match G to A, what I'm doing is I'm providing the A and the G and requesting a clean and sorted matrix. So it's going to give me 2 equivalent matrices, the A clean, what I call, and the G clean. They are both matching perfectly one on top of each other. And that's very valuable information. So it says here, all 644 individuals on the matrix G are on the matrix A, which is very important, because that means if you genotype something wrong or you have errors on your IDs from your genotyping, the names might not match what you have in your pedigree, or there's individuals that you don't have in your pedigree that you need to bring in. So that creates a bit of an issue, okay? So, and on the other hand, it says there's 1,390 individuals that were not present on G, but they were on A, which makes sense because our A matrix has the full pedigree. So that's nice. The dimensions are both the same, both of the clean, and I just want to show you 2 bits of both of the matrices that are now matched perfectly in order. You have again 1 and 0.5, 0.97, and you have a 0.38 for the relationship instead of being 0.5. We have 0.5, 0.5 here, which is 0.45, 0.48. And those values are slightly less than 0.5 and 0.25 because we have that shift. So we'll do a little bit of things. This plot that comes from that function is probably the most important plot you can have. This plot is matching the A to the G, so the A is here and the G matrix is here, and you can assess inconsistencies. For example, individuals that were declared full sieve, they might have values on the genomic data that are around 0, which means they are not full sieve. That's an error on the pedigree, and the pedigree needs to be corrected. On the other hand, you might have individuals that the pedigree says they're unrelated, and then maybe they have around values of around 0.5, which means the pedigree, again, it's failing to declare those to be related, and it might be because of pollen contamination and so on. Now, this graph presents our opportunity to correct as much as we can our pedigree file and also detect inconsistencies on the DNA. We cannot say where the problem is because sometimes the DNA could be that some files got swapped or something that happened that we need to assess. So it's hard to blame somebody, but it's very important to use A and G to clean both things. And we generate another matrix that we call RM, which is actually tells me it's the same matrices but put next to each other in columns. So you have here, I just did a filtering of all those records where the difference between the value from the A matrix and the G matrix is bigger than 0.20. And in this case, I have a group of individuals that are being declared to be full SIPs, and it happens that their value on their genomic matrix says they are half SIPs. Could be a potential pollen contamination, or it could be an error on the pedigree where we're assigning the mom or the dad incorrectly. Some of those can be corrected, others cannot be corrected. So that's very important, and I do recommend a lot of investment time on that, okay? So then now I can move into the alignment of the matrices. So in this case, what I'm going to do is I'm going to move the G towards A. And when I do that, there is a number here that we use to denote the quality of the matrix. You won't see too much of a difference between these two matrices. But what you will see is that the numbers shift a little bit closer to 1 with the new aligned matrix. Now we have a 0.38, it moved to 0.41. And in reality, the main thing I really, you will see this graph will be centered on the line. And one of the other things that it's important is that we'll have fewer records. We have older records that still bring us some issues like more than 20. This is the original one. No, this is the new one. And we have no records with problems of bigger than 0.25 and 90 records with problems and difference more than 0.20. So if you remember our off diagonal originally from the unaligned matrix had this. Now with the aligned matrix, and please notice the shift that you're going to see, the lamp here is going to move, and you see 0.25 and 0.50. So now we have a good matrix to use. Okay, so now I feel I'm ready to use this genomic matrix. And then again, I have a matrix with 644 records, and I have a phenotypic data that 861. So this function, match kinship to pheno, what it's going to do is going to drop from the phenotypic data those records that are not found on the genomics data. Okay, so that's important again to try to have everything kind of like aligned very well. You can leave the records on the genomic data that are more, but every individual who is in the genomic data, no, every individual who is in the phenotypic data has to be present on the genomics data, which happens in this case, but you can have more elements on your genomic matrix. So in order to do my analysis with ASRemol and any other packages, I need to make sure I can get the inverse of this original G-aligned matrix that I have. Okay, notice that in this line, the only thing I did is a subset of the phenotypic data. So there's many ways. This function solve from R is not the only one that we can use to do this, but there's many ways to obtain this inverse, and we came up with one that is a bit more robust, and we came with some statistics that tell me if that inverse is ill conditioned or not. The conditional number is very good in both of these cases. So now I have a genomic matrix that can be used directly in ASRemol. Okay, so now I feel I'm ready to use ASRemol to do my analysis. Okay, and again, this has been kind of like a long process to try to get the matrices ready to do my analysis and trying to get this G aligned, but it's very important because it avoids conflicts downstream. Okay, again, you can use different algorithms, you can have different data, but the process for all of those cases are very similar, where you have to do some quality control, you have to get the matrix, assess that everything's okay. In this case, I assess it with the A, and then get to the inverse. So I'm going to use ASRemol now. I do have my license for ASRemol, and I'm going to use the subset, the pheno subset that is matching the ones that I have on the G matrix. So actually the dim of pheno subset is 644 records. Okay, I have 644 records. All of those are being genotyped, 644 individuals, so it's one-to-one, no problem. For ASRemol and many of the libraries for linear mixed models, we need to make sure the genotype is as a factor. Actually, here as we have the genotype as a factor, we won't use this, but then we have our response variable. And again, just to check that everything is okay. So now I can use the ASRemol. If you're familiar with ASRemol, you have this command ASRemol, that function. Fix is the argument to denote the response variable and any other fixed effects that you might have. For example, I might have block or something. In this case, I don't have any other random or fixed effects. And then the random component equals tilde. And with this function, we say, please use genotype factor. And to the genotype factor, assign the G inverse matrix, which is the matrix I just calculated here from the align matrix that I'm happy with. So this align matrix is inverted, stored in a sparse form with some attributes. I do assign that directly to ASRemol. And if I want to add heterogeneous errors and some other things, everything will go okay and no problem. So now I run my GBLUP, and this again is just directly genomic matrix. It's a direct genomic selection model, 644 records. I'm going to get my GEVBs. You can see ASRemol runs, no big warnings. And finally, I have some variance components that are estimated. I can do a residual plot. And in the residual plot, we can assess for outliers or something we need to do, transformation, etc. And then we have a function which is called bpredict to give me a heritability. This is a genomic heritability. Again, there's big debate about genomic heritability or regular heritability, but I'm just taking the variance of the genetics component divided by the sum of those two terms, and that gives me 0.45. And finally, I can assess my GEVBs with my genomic values, and I can rank them, I can view them, and I can select the one with, in this case, is yield. So the one with the highest positive value is the one that will give me the best breeding value for, in this case, kind of like a GCA. So that's fine. And that's my GBLUP analysis, but I haven't used my genomics data. Okay, so what we're going to do is I'm going to go to this slide where we describe the single-step GBLUP. And I know many of you are aware and know about it, but I think it's a very useful tool for us as plant breeders, and I say it mainly for the tree breeders, where we take a long time to generate phenotypic data. So generations that take 7 to 10 years in some of the cases. So it is a methodology that comes from animal breeding. And again, it will combine the pedigree and the molecular data. It's called single-step GBLUP because originally in animal breeding, they did 2 steps. And now they combine it into a single step. For us, single step sounds very strange. I prefer to call it HBLUP for hybrid BLUP because it uses both of them. And you can see here, you can obtain an H matrix that takes the A matrix, and then it takes some of the G information, the genomic matrix, replacing the portion of the A matrix of those individuals that we have genotypes, so you can subtract. But it's a bit more complicated than that, and you can see these complex expressions here, because the main thing that happens is now I'm saying these 2 individuals are related now, and let's say they are cousins, distant cousins, but we didn't have that from the pedigree, which means somehow the parents are related, and we need to fix the relationships of the parents. And this is what it happens with this AA1 term, or in this case, H11 term. And that obviously is good, because then it uses the information from offsprings that are related. That means the parents have to be related, or grandparents have to be related, and fix everything. So it has quite a good information on that. Now, instead of going to this intricate matrix algebra, it's a lot easier to just work directly with the inverses. And you have these inverses here with lots of zeros and mathematically much easier. But there's 2 parameters here that are tau and W, which in reality what they say, and think about this, if tau and W are the same, I can say I want 90% G and 10% the A inverse. So what you're doing here is you're saying replace on the A inverse the information from the genomic matrix, but it still keep 10% of the A matrix because there is some information on the A matrix. And this is respecting that, which is nice. But also when you have errors on the G matrix, it kind of like smooth out those errors and it still gives value to the A matrix. So that's why it's called kind of like a hybrid. Obviously, everywhere else only has A information or A minus information. So then you will only use that. Okay, and that's what I'm going to show you. We can use a parameter lambda, which is actually with tau and omega equal, or we can specify these two parameters independently, because sometimes some adjustments make the, or some values slightly different. Let's say 90% here and 12% of A inflates a bit of the diagonal, and then that makes the inverse a bit more stable, and then you can get the results. Okay, all of that we implemented in the ASRgenomics, and again we'll be using ASRemo to do that. Okay, so it is very simple. One of the things that we need is we need the inverse of the G aligned, which is our 644 by 644 matrix. And we can see it a little bit here. It's just an inverse matrix that doesn't make too much sense, but it's based on molecular data. We know it has a good determinant. We know it's invertible, obviously, because we got it, and it's very stable. Now I take that inverse of the G and I take the A matrix and I can calculate an H inverse. Now notice that I'm setting up lambda to 0.9, which means 90% G, 10% A. I could put a 99 here, or you can eventually play with different proportions and even look at your log likelihood value to decide which is the proportion that gives you the best or the highest likelihood value. And from there, make the decision. It is very important. Sometimes the A matrix is a lot more information than you think, because maybe the A matrix has been checked, the pedigree validated, you have a good record of it. And the G matrix, let's say, is only based in 1,000 markers with potential mistakes. So then it's good to combine it with the A matrix to make that G matrix a bit more robust. And sometimes it's the other way around. The G is very good, and we know the pedigree is very bad, so we try to not use too much of the A matrix. But in any case, you fix A matrix as much as you can, because A is being used directly on the calculation of this H. So bad A, like they say, garbage in for A, is going to make that G matrix a bit of garbage, and you're not going to improve your analysis. So this H inverse, again, it just looks a bit odd for us. It has 1,390 individuals that are not present in the G inverse. So again, this is the parents and grandparents, plus those 200 and plus individuals from the offsprings, which we have genotype, but they have no information. So one thing we can do is we can explore the H matrix, and I like to explore the H matrix. Now, this takes a bit of time to calculate because again, as I showed earlier on the slides, you have to do some matrix multiplications and so on. Why do I like to show this H matrix? Is because we can see what changes have happened. So notice here, and this is also very good to assess potential errors on the pedigree. The parents, this is a group of a few records of parents 25 to 30. They have value of 1 and 0 on the off diagonal. And you can see some parents seem to have some relationships. 5%, 5%. There is a 6% here. So this is not bad. There is some maybe distance cousins and so on parents, even that originally we know they were unrelated. And this is again because some of the grandkids are related, but they are not declared on the pedigree. So let me just show you another set of parents. Actually, these are the second generation. So these parents are half-sifts, and you can see again a 0.22, a 0.22. So they're being corrected a little bit with the H matrix because those parents have genotyping information through the kids. And again, so we are allowing the parents to have some sort of Mendelian sampling. There is a 0.16 here, which is quite different than that 0.25. So maybe something happened. And finally, we have the A for the individuals. This is the offspring. and how the A is once we have this 90% G and 10% A. So we have, again, some relationships 0.5, 0.5. So they are close to 0.5. This one is a bit far. And there is a negative value here, which is very close to 0. So effectively 0. So that's nice. We can do some diagnostics of this H matrix as before to see if we have duplicates, if we have some inconsistencies, if we have values that are too weird, like high levels of inbreeding or so on. Now, this diagonal looks a little bit more strange than before because we have these two sources of information. It's not as smooth. And you see something similar with the off diagonal where you see sort of these peaks where's the 0.25 and 0.5 relationships, because again, we are using everybody who is 0.5 or 0.25 even on the actual pedigree. So it doesn't look too nice, but it's not much of a problem. So finally, I take this H inverse. I want to calculate my H inverse. So you provide the A, the G inverse, the lambda value, and I show you this one. I'm asking for that to be in a sparse form to be used directly on ASRemol. So fine. And now notice I'm not using my subset data. I'm using my phenopine. So what is the dimension of my phenopine? It is actually 861 records. And this is where I like to present. I could have, and actually the dimension of the H matrix, which I'm going to do here, it is 2034. So eventually I could use this H inverse that originates from an H that has phenotypic data on 2000 individuals. I can go back to my first generation of trials, know that the parents are there, use their phenotypic records, that will be the grandparents and the parents and so on. And if you think about it, you're not moving too far away from your typical pedigree-based analysis that you did before you had genomics data. Now you are using both of them together in this single-step GBLUP. So I'm ready to fit my model in ASRemol or any other software of your choice. Notice it will take a little bit longer, of course, because this matrix now is much bigger, 2000 instead of 644. But the logic is the same, H inverse dot spar. If you remember what I did in my GBLUP, I used G inverse dot spars. That was 644 by 644, and this is H inverse. If you were doing a multi-environmental trial analysis and so on, you can add site, you can add an interaction with a factor analytic, you can have heterogeneous error, you can do whatever you want because ASRemol allows you to have that flexibility on the same way that you will have done your pedigree-based analysis. It's going to take longer, it's going to require more computing resources because this H matrix is a lot more dense in data than your originally pedigree matrix, okay? So, but as you can see, in this case, it didn't take too much time. So my residual plots are going to be a bit different because remember now I have 800 and so records of phenotypic data. The variance components are going to change 134, 134, 124. I think before I had these results, 117, 138. Now, the main thing you will see is that your standard errors on the new analysis, if your phenotypic data is good and if your pedigree is good, are going to get reduced. Potentially, you might have more inbreeding, more heritability, but that depends on the quality of your data because it could go either way. So I'm calculating the heritability is 0.51. Again, it's a little bit higher than before. You can see these variance components went to 1.34 instead of 117. So am I happy with this? Yes, I am very happy. It still runs. I didn't have to manipulate the H matrix. And now I have my genomic breeding values, but in one sense, I cannot call it genomic. They're hybrid breeding values because I'm combining both things. So you can see this dimension of HGEBB is 2000 records because everybody who was defined on the H matrix, in this case, the H inverse, will have a breeding value. And you can see here some of the parents and some of the kids, and you have the standard errors. And notice the parents, we'll have a standard error that possibly will be much higher. In this case, it is in general, because they are not being genotyped and the relationships are poorer with the rest of the data, which is in this case, the groups of offspring. In some cases, they seem very nice because probably this grandparent has lots of parents and those parents have lots of grandkids. So again, it will have probably more links with the information, and you can see the standard errors of the rest are very good. So I'm just going to do some small manipulations. These two data frames are very different to what I do. For example, I can do view HGEBB, and then these will be the breeding values, and I can rank this from the largest to smallest and actually select, and the first best individual happens to be a kid, but the second one happens to be a parent, not a grandparent, a parent. So, and then this individual is probably not genotype. So that's very good. It's not genotype, but it looks to be very good because the offspring is very good. So now I can go and say, oof, do I have it? Should I use it again? Do I have pollen stored somewhere? There you have another one here, and so on. So it brings that kind of nice option. So on the next line, and just to show you the last few bits before the hour is up, this is just our code where I'm just extracting the names of the genotypes and merging them into a single data frame that I call TT. So where I can compare one-to-one those individuals who were genotype and fitted for the GBLUP against those who were fitted with the HBLUP. So just trust me on my code. And here I have the X's represents the old analysis and the Y's represents the new analysis. So let me just show you here. So this X's represents the GBBs, and you have this individual have a GBB of 0.71. The new analysis gives me a 0.81. The standard error was 674, here is 673. So you can see a little bit of a drop on the standard errors on the individuals from before and now. And this is nice, and it makes sense because we have a portion of data. This drop could be a lot more if we have more information from those parents. More information, more individuals phenotype, and then obviously consistent with good heritability, this will drop. But the changes on values are relatively minimum. Again, depends on how much data you have. Now, one of the things that I put here in the summary that I find very interesting is that the mean of the GBLUP analysis was 0.002, so almost zero. Because it's my reference population, I don't have anything. I don't have any selection. I don't have anything I've done. It's just my G-align. So it makes sense that the mean is around zero for the breeding values. But the breeding values of the new analysis are 0.15, which is bigger than 1. So everything will be shift 0.15 more or less. And why is that happening is because I'm using the A matrix. The A matrix knows there was a selection and some of the poor parents are not present on the latest generation. So that bias that is produced there is going to be corrected in this particular case. So it is important because not only does that, it corrects. You can see the correlation. Well, this is not the correlation, but if I can calculate the correlation between these two, I'm suspecting it'll be 99 or 0.9969, and you can see here there is still a bias, but you can see the changes on between the two analyses for those individuals is minimum. It again is just respecting that there is some pedigree, and it's the time percent of the A matrix. So this is, I'm quite happy with this. It just kind of brings all the information, and this is what I wanted to illustrate. So I have a few more minutes. I just want to talk about the final things. Again, this is an illustration of the tools. As I say, you can use other tools if you want. But I do think, and this is part of our consulting too, there is information that most breeding programs will have. Plant breeding programs, animal breeding programs, tree programs particularly. So it doesn't make any sense that we don't use this pedigree information. And in some programs, it is there and it's never been used to assess or to help genomic models. Remember, you can also play with the proportion of one and the other, depending on which you trust more. Pedigree information is valuable. And again, if we don't have too much genomic information because we are starting to build our breeding program with genomic tools, then it makes much more sense to try to use the pedigree. Now, we can also use this single-step GBLUP or HBLUP to kind of tweak up our strategy. Maybe not all individuals are going to be genotyped, only a portion of them. And let's say, for example, you might want to genotype what you consider to be the top 50% of your individuals from an assessment, of phenotypic assessment with a 50K chip, and the other 50% you only do it with a, let's say, 100 SNP chip, because you only want to do pedigree reconstruction and use their pedigree. But they are phenotype, so you have information, and you can assess those and combine in a nicer way with cheaper resources. Now, genomic data is not going to replace the pedigree data immediately. At some point, it will replace it because we will do everything with genomic data. But at the moment, because we are starting with most programs on genomics data, we move slowly to it. So as we don't have the shift, and as we understand how genomic data works in the specific breeding program, I think the pedigree data has a lot of value. Okay. And we still have to rely on the pedigree in the future, I think, just for pedigree, parentage reconstructions as in some tools. But I still think the pedigree will help fix issues, correct what happens with the genomic matrix or the other way around. So it has value. And one thing that we discussed with some colleagues is that the A matrix has something that the G matrix often lacks, which is a good estimation of the inbreeding based on a base population. So if you want to track down the historical inbreeding, still the pedigree is the one that does the best job. And some of the relationships are lost of diversity and so on. So I think we still need to go a little bit back to the pedigree. And the last thing I want to mention is that pedigree is not easy to manage. Even if it's reconstructed or not, it requires always a good database system. You have to go back to it, you have to tweak it, you have to find mistakes, come back, names, and so on. But I do personally think it's a good investment because as we see with the HBLUP, it brings a lot of good things. So I have 1 minute, so I've timed this perfectly. Again, thank you for joining us. I know I talk a little bit quick, but you will have option to access the code and access the slides. If you need our help, and particularly my help, we offer consulting. We have support for our software. If you want to play with this code, also please contact us. We are really there to help you and to tell us what your needs are and so on. So I want to thank your time.
Fabulous. Thank you, Salvador. Incredibly informative, as always. We'll all be dreaming about matrices tonight. We have one question in the chat. I know we've actually gone over time, but I am just going to pose this one question. I'm not sure if you'll be able to answer it today, but obviously we'll come back to you. A question from Mohammed Iftikhar. Excuse my pronunciation again, Mohammed. And he's asking, what if we don't have the actual pedigree data, or can we get pedigree data if our selection history or pedigree, i.e. series of parentage, is given? Can we use that data and convert it into the data that you used?
Yes, the answer is yes. There is some breeding programs that work with pedigree reconstruction. So they don't know, you might know what parents you use, you might genotype the parents, but you don't necessarily know the actual pedigree. And it's a good strategy because you can obviously use SNPs that have, chips that have much less SNPs. When the programs get much more generations, gets a bit more complicated to reconstruct the pedigree. But remember, the pedigree is expected values. So yes, there is information in there. So I think the key is to use a combination of both.
Lovely, thank you.
Good question.
On that note, perfectly timed. Thank you once again, Salvador, and thank you everybody for joining us. I hope you found that informative. We will be following up with the recording and further details. So hopefully we'll all be in touch, or we'll see you at the next webinar. Thank you and goodbye.
Thank you very much for joining.